Hello everyone. Yeah, in this video we will discuss about a type of division which is called mitosis. Children, this is very important topic for your need purpose as well as for your examination purpose. So listen it carefully and watch the video till its end. Okay, first of all you should know what is mitosis. Mitosis is that process of cell division in which one cell divide to form two daughter cell. That is called mitosis. Now, the two daughter cell which are formed, they have same number of chromosome as were there in the parent cell. Suppose parent cell have 46 chromosome, so we got these two daughter cell. These daughter cell have also have 46 in this, other will also have 46 chromosome. So we can say that mitosis is that type of division in which from one cell two cells are formed and both the cell have same number of chromosome as were there in the parent cell. And because of this, mitosis is also known as equational division. Right? Very important this is that which type of division is called equational division. So mitosis is called equational division. What is reason behind it? Because the daughter cell have same number of chromosome as were there in the parent. Now further it is written that it is that type of division in which chromosome replicate and become equally distributed, but quantity and quality in the daughter nuclei so that it remain the same. As you know children, in our body there are two type of cell. One type of cell are called somatic cell, which are also called body cells. Other type of cell are called sex cell. तो जो माइटोसिस होती है it take place in somatic cell of a body. Sex cell are present in sex organ, like in the female they are present in the ovary, in the male they are present in the testes. So besides sex cell, rest of all the cells of a body they divide by mitosis. Now we will discuss in detail about mitosis. So mitosis has two steps actually. There are two steps involved in the process of mitosis. First is called karyokinesis, second is called cytokinesis. Now what they are actually? Okay, so mitosis has how many steps? Mitosis has two steps. First step is karyokinesis. Karyokinesis. So as the name indicates, karyo always means nucleus. You should know about it. Karyo means nucleus and kinesis means division. So we can say that karyokinesis is division of nucleus. So whenever mitosis occur, the first step is karyokinesis, in which the nucleus of the cell divides into two nuclei. Then afterwards cytokinesis take place. Cytokinesis means cyto means cytoplasm, kinesis means division. So now we will discuss in detail first of all about karyokinesis. What happen in karyokinesis and how do it happen actually. So if we talk about karyokinesis, then in karyokinesis there are four different stages. And what those stages are? The first stage of karyokinesis is called prophase. The first one is called prophase. Second one is metaphase. The third phase is called anaphase. And the fourth one is known as telophase. So there are four phases: prophase, metaphase, anaphase, telophase. Now one by one we will discuss about each of them, what happened in all of these stages. Okay, so you can have a look at the diagram here. This is the cell. This is an interphase cell. You know that when the cell has entered in the interphase, then what has happened to the cell? In the interphase, cell का हर एक organelle almost duplicate हो गया था. You can see here is centriole. तो centriole भी duplicate हो चुके हैं यहाँ पे. And these are the DNAs here. These are the DNA. DNA की replication हो गई है. Amount of DNA is double in this particular cell. ठीक है. Now this interphase cell will enter into the first stage, which is prophase. Now carefully listen what happened in prophase. प्रोफेज में क्या हुआ, ये जो क्रोमेटिन फाइबर थे इंटरफेज में, दीज क्रोमेटिन फाइबर वर अनकॉइल्ड. तो प्रोफेज में उनकी धीरे धीरे कॉइलिंग होना शुरू हो गई एंड दे स्टार्ट विजिबल एज डिस्टिंक्ट chromosomes. And the next change take place in the centriole. The pair of centrioles start moving away from one another, and around each centriole ester rays appear. Ester rays, you can see they are appeared there. और ये जो centriole है, they start moving away from each other, and in between them these spindle fiber appeared. And further, this is the early prophase. And then in the later prophase, in the mid prophase and late prophase, what happened? You can see here what has happened in this cell. In this cell, the chromatin fiber has become sufficient thick and they are visible to us as chromosome. और न्यूक्लियर मेम्ब्रेन धीरे धीरे डिसअपियर हो गई है. Earlier there was nucleolus here. Now nucleolus was started disappearing in the early prophase. You can see here it started disappearing here. यहाँ धीरे धीरे डिसअपियर होना शुरू हो गया और यहाँ पूरी तरह से डिसअपियर हो गया है. तो व्हाट वी विल से कि व्हाट हैपन इन द प्रोफेज, न्यूक्लियर मेम्ब्रेन स्टार्ट डिसअपियरिंग, nucleolus disappears, chromatin fiber become coiled, thick and dense and start appearing as chromosome. The centrioles start moving away from one another and ester rays are formed around them, and in between the separating centriole the spindle fiber are formed. Clear? Now this is the diagram of late prophase. At the end of the prophase, what happened? When the prophase ends, till then the central part which was occupied by nucleus, that is fully disappeared because the nuclear membrane is broken. और ये जो centriole हैं, they reach at the opposite end of the cell, or the center of the nucleus is occupied by these structure which are called spindle fiber, and chromosome come to lie over spindle fiber. This happened in prophase, at the end of the prophase. Now there is metaphase. Next stage was metaphase. In the metaphase, what happened? In the metaphase, as the centriole have reached at the end, the center of the cell is occupied by the spindle fiber, and all the chromosome come to lie in the middle of the cell, and they form a single plate, and this is called metaphasic plate. So in case of mitosis, a single metaphasic plate is formed. Clear? Afterwards, the next stage after metaphase is anaphase. Children, from metaphase, one question is asked in NEET that if you want to study chromosome, then what is the best stage to study them? So it is metaphase. Why? Because in the metaphase stage, the chromosome are highly condensed and coiled, and they are easily visible to us. So the best stage to study chromosome of a cell is when a cell is in its metaphase stage. Now, what happened further, that we'll discuss. Next phase is anaphase phase. In anaphase, actually what happened, this is a chromosome. Actually a chromosome is drawn here like this. So what these two parts of the chromosome shows us? We know that this particular cell, before entering into mitosis, it has gone through interphase. और interphase में one of the stage was S cell. वहाँ पे क्या हुआ था? If this is the DNA, so that DNA has replicated. One more DNA like that has been formed. So on one chromosome, the amount of DNA has been doubled. That's why when this chromatin fiber coiled and condensed, it took this kind of shape. So it's one part from here to here. That is one DNA, we can say, and the another part is its replicated form of DNA. And these two halves of the chromosome, one is this one, another is this one. I can show it with a different color. The two halves, one is this full part, this particular part. One is this part, and this part. This is called one chromatid, and another was this one. This is the second chromatid, like this. So what it is actually? It is actually the replicated DNA. This was a single DNA. एस फेज में उस डीएनए के जैसा ही एक और डीएनए बन गया था. दैट्स व्हाई वी हैव शोन क्रोमोसोम लाइक दिस इन मेटाफेज स्टेज. नाउ इन एनाफेज स्टेज व्हाट हैपन? In anaphase stage, this DNA gets the two DNAs get separated from each other. And how do they get separated? Have a look here. This was one of the DNA here in the chromosome, and the another DNA was this. This was the second DNA. Now, what happened in anaphase? In anaphase stage, these two DNAs get separated from one another. One of the DNA start moving towards one end, like one of it start moving towards one end of the pole, one pole of the cell, and another one start moving towards opposite pole of the cell. So this happen in anaphase actually. So what is happening in anaphase? In anaphase, one chromosome, this whole was a single chromosome. So in anaphase actually, a single chromosome split and one of its chromatid move towards one end of the cell, another chromatid start moving towards the opposite end of the cell. So actually what is happening in anaphase? It is shown further here in the next diagram also. Yeah, you can have a look here. This is the late anaphase they have shown. What has happened at the late anaphase? At the late anaphase, the chromatid of each chromosome has reached at opposite end. Clear? तो in anaphase actually what was happening? क्या हो रहा था anaphase में? In the anaphase, the amount of DNA which has been doubled in each chromosome, that is reduced to half. It is not there कि chromosome is number is reducing to half. Now I will explain it again. Anaphase में क्या हुआ? Amount of DNA is reduced to half. This is very very important question for NEET purpose, that what is the basic function of anaphase stage of mitosis. So anaphase stage of mitosis में DNA is distributed equally in both the daughter cell. Clear? Now further what happens? What happened at the later? Okay, this was late anaphase. So at the end of the anaphase, towards the later anaphase, what happened? The chromosome separates. Each chromatid of the chromosome start moving towards the opposite end. They have reached at the opposite end, and the spindle fiber which were in the center, they start disappearing, and the chromatin fiber, they reached at the end. Now the next stage is telophase. Now what happened there? As you can see here in the telophase. In the telophase, the chromatids has reached at the opposite poles. स्पिंडल फाइबर पूरी तरह से ऑलमोस्ट डिसअपियर हो जाते हैं. वेरी फ्यू आर persisted in the center. And this telophase is exactly reverse of anaphase, prophase. Prophase में जो हुआ था telophase में उसके opposite होता है. So in the telophase actually what happened? In the telophase again the nuclear membranes start appearing around it. Here also the nuclear membranes start appearing. In prophase it has disappeared. Nucleolus has disappeared there. यहाँ पे फिर से nucleolus appear होना शुरू हो जाता है. And one more thing has happened in the prophase. In the prophase, the chromatin fiber which were uncoiled, they start coiling and thickening and start appearing as chromosome. तो यहाँ पे क्या होगा? Reverse of that. यहाँ पे we have a chromatid. So this chromatid start uncoiling and again become the chromosome here. So suppose we started with a cell where there were two chromosomes. Clear? So there were two chromosomes in the cell. This was a chromatin fiber. This was another chromatin. Two chromosomes are there. So this cell enter into interphase. Interphase में क्या हुआ? What happened in the interphase? Okay, once again I'll draw it. I have a cell. This is the nucleus I'm drawing. This is one nuclear, one chromosome. This is another chromosome. Now this cell enters into interphase. इंटरफेज में व्हाट हैपेंड, इंटरफेज में इसका जो डीएनए था वो रेप्लिकेट हो गया. आई विल शो रेप्लिकेटेड डीएनए विद सम अदर कलर. दिस इज रेप्लिकेटेड डीएनए, this is replicated, and that was the original DNA. So this has happened when this has happened in the interphase. Now the cell will undergoes mitosis. अब इसमें mitosis होगी. अब जब mitosis होगी, so what will happen, how the mitosis will happen? This is the nucleus, this is the DNA, and this is second DNA. हर एक DNA के ऊपर क्या हो गया है, this is the second part of the DNA, second DNA replicate हो गया है. Now दिस पर्टिकुलर सेल विल अंडरगोज प्रोफेज. प्रोफेज में क्या होगा, जैसे ये प्रोफेज में एंटर करेगा, ये जो क्रोमेटिन फाइबर हैं ये धीरे धीरे थिक होना शुरू हो जाएंगे. एंड दिस विल अपीयर एज अ सिंगल क्रोमोसोम, बट दिस इज वन हाफ ऑफ द डीएनए, एंड दिस इज अनदर डीएनए, रेप्लिकेटेड डीएनए. सिमिलरली दिस इज अनदर क्रोमोसोम. इसके भी दो पार्ट शो किए हैं विच आर शोइंग द रेप्लिकेटेड डीएनए. नाउ इट विल एंटर इनटू द मेटाफेज, जिसमें सारे स्पिंडल फाइबर पे अरेंज हो जाएंगे. फिर एनाफेज में व्हाट विल हैपन, इन द एनाफेज नाउ as in the metaphase, what will happen, this is the spindle fiber, one of the chromosome will come to lie here like this, another will come to lie over another one like this. Now this will undergoes anaphase. What will happen in the anaphase, in the anaphase stage, now the replicated DNA will start separating from one another. Means one half of the chromosome, this half of the chromosome move toward this side, another half will move toward this side. So what is happening here actually, this is one half, this is another half. This is about second chromosome one half, this is about second chromosome one half. अब ये टीलोफेज आएगा. टीलोफेज में न्यूक्लियर मेम्ब्रेन बनेगा. This will again uncoil, will form one chromosome, this will form second chromosome, this will form one chromosome, this will form second chromosome. So now we have, here we have one cell nucleus, here we have another nucleus. How many chromosomes within this nucleus? 1 and 2. How many within this? 1 and 2. This was the parent cell. How many chromosome parent was having? 1 and 2. So as parent is having 2 chromosome, जो daughter nuclei बने, they also have 2 chromosome. That is why mitosis is called an equational division. So this is how nucleus divides. So finally, after the division of nucleus, 2 nuclei have been formed, and then the cytoplasm starts dividing. And the division of cytoplasm helps in equal distribution of cell organelles in both the cells, and finally we get 2 cells. So this is how actually karyokinesis take place. Thank you.